Hello, my name is Sierra Williams, and this research was conducted with the guidance of my advisor, Dr. Jennifer Grindstaff. Our study evaluated long-term consequences of neonatal antiviral response on behavior in the zebra finch. There is considerable covariation between immunity and behavior, as pathogens can influence behavior and behavior can influence pathogen acquisition. Early life experiences may serve as a potential mechanism for this apparent covariation, as the neural circuitry that comprises the adult brain is incomplete during this period. Thus, the neonatal brain is particularly vulnerable to extrinsic and intrinsic challenges. Exposure to viral pathogens initiates a cascade of potent inflammatory cytokines that dominate the immune response, which are critical for viral clearance, as neonates have a limited ability to synthesize antibodies. The energetic cost of inflammation causes lethargy and anorexia, which can impose nutritional constraints on developing young. In the central nervous system, inflammation exerts changes on neural circuitry, potentially leading to synaptic disorganization. These short-term effects may result in long-term consequences, like altered growth and changes in adult behavioral phenotype. The objective of this study was to evaluate potential long-term effects of an antiviral immune response on behavior, specifically learning and neophobia in zebra finches. Understanding how the neonatal immune system affects other life history traits would bolster our understanding of immune-mediated trade-offs. For our immune challenge, we used the viral mimetic Polyinosinic-polycytidylic acid, or Polyic. Polyic is a synthetic double-stranded RNA virus and a toll-like receptor 3 agonist. Birds were challenged on post-hatch day 5 with either Polyic, saline, or were simply handled as a no-injection control. At 6 months post-hatch, birds were assessed for neophobia and learning proficiency. We used a food-driven novel foraging board with 4 successive levels of increasing difficulty. As the levels increased, lids were used to make it more difficult for birds to access the wells filled with seed. To pass a level, a bird needed to eat from 2 wells. If a bird failed a level, they had to go back to the previous level. Birds were given 18 attempts to pass the entire assessment, which meant completing level 4 twice consecutively. They could theoretically do this in 5 attempts. Neophobia was assessed using a novel object trial. Anxiety-related behaviors like calling, perch hopping, and side flying were quantified during the 10-minute trial. We used a generalized linear model with transformed data to assess the effect of treatment on behavior during the neophobia trials. Ultimately, we found no support for treatment as a response variable. These results may have been due to the timing of the initial injection. A study with mallards challenged with sheep red blood cells during different developmental time points only showed changes in neophobia during middle and late developmental periods, between 8 to 15 weeks of age, after the period of maximum somatic growth, but prior to acquiring adult plumage. We used a generalized linear model with a Poisson distribution to examine what factors might influence how many trials it takes an individual to pass a learning assessment for those individuals who did pass the learning assessment. We found the model using treatment, pretrial weight, and sex as 3 independent fixed effects was the best model. What I want to draw your attention to is that Polyic and saline-treated males and Polyic and saline-treated females are nearly indistinguishable from each other. Additionally, the effect of weight appears to be consistent across sex and treatment. Thus, there are 3 independent effects going on in this analysis: sex, weight, and treatment. With that in mind, I was curious if pretrial weight was affected by treatment, and if so, was this a carryover effect from early life? As it turns out, treatment does affect pretrial weight. And this also appears to be a carryover effect from the fledging period, where there is a significant difference in the fledging weight of injected individuals, which encompasses saline injected individuals and poly-IC injected individuals, from non-injected individuals. However, there isn't a significant difference in weight between the saline and no injected group in adulthood. So why is there a difference in learning proficiency? Well, research in the area of neonatal pain might provide some insight here. Neonatal pain has been linked to adult hyperalgesia, an increased risk of poor health outcomes, disorganization of the central nervous system, and altered adult behavior. It could be the saline injection alone may have served as a sufficient pain stimulus to elicit similar pathologies as the poly-IC injection. I suspect the weight trends seen during the fledging period may be due to flight-related trade-offs. That is, these individuals may be forced to allocate more resources toward flight feathers due to nutritional constraints imposed by the initial inflammatory response. These preliminary data suggest a no-injection group may be a potential consideration for future early life studies. I would like to take this time to thank my incredible funding sources, as well as my amazing undergrads and fellow lab mates for their assistance in this research. And thank you to the Animal Behavior Society for having this conference.